Afternoon, everyone. Welcome to the 16th edition of Sun and Science, which is the summer version of our Soup and Science event, which we run otherwise in September and January. So today we will have four excellent speakers who will present their research to you in short 3 to 5 minute talks. I'll let you know who they are. So we will start with Professor Christopher Thibodeaux from the Department of Chemistry, followed by Professor Yves Lee from the Department of Physics. Then we'll have Isa Dang from Physics Matters and Astro McGill Outreach Group. And our last speaker today will be Professor Wayne Pollard from the Department of Geography. In case you've joined us before, usually after the talks, we will divide you up into breakout rooms. That will not happen today. So the discussion, there will still be a discussion, but it will take place in the main room where you currently are. Okay, and I think that's all that's needed in terms of introductory remarks. So let's just get started. And I would like to introduce our first speaker, Professor Christopher Thibodeaux from the Department of Chemistry.
Okay, everyone. Yep, I'm Professor Chris Thibodeaux from the Department of Chemistry. Thanks for the great introduction, Axel. And today I'd like to talk to you guys about how my lab is using mass spectrometry to do so much more than just to measure the molecular weights of molecules. And so this is a picture of the mass spec instrument that we have in our lab. And I'll briefly run through how we're using this powerful analytical platform to study peptide antibiotic biosynthesis, and then to study the biophysical properties of the enzymes that are involved in making these peptides. And so first, one of the main areas of focus in my lab is to understand, we study this group of enzymes that are involved in the biosynthesis of antimicrobial peptides. And so what these enzymes do is they take a linear peptide and then they install a series of post-translational modifications, a series of chemical modifications into the peptide. And what's really remarkable about these enzymes for us is that these enzymes are multifunctional, and they often have multiple active sites, and they catalyze different chemical reactions, and they do this in an iterative fashion. So this one enzyme will catalyze about 20 different modifications into this peptide. And what's really intriguing is that the precise identity of the modifications is very important for giving the peptide its biological activity. And in this particular case, this peptide is an antibiotic. And so we're very interested in how these multifunctional enzymes control their multiple activities in time and space in order to make very specific final products. And so, as I mentioned, we use mass spectrometry to characterize many different aspects of the structure and function of these enzyme systems. And so mass spectrometry is very useful for these enzymes because after each round of chemical modification, the mass of the peptide changes. And so we can use mass spectrometry to detect all of these intermediates. We can basically inventory at any given time in the reaction which set of intermediates are present. And moreover, each one of these little signals that's numbered on the graph here corresponds to a different intermediate. And in our instrument, we can select each intermediate that we're interested in, and we can blast it apart into tiny pieces, and we can map out precisely where all of these modifications are located. And we can also, by quantifying the intensity of these signals, we can fit this data to kinetic models to really extract a lot of detailed and quantitative information out of these multifunctional systems. And so one of the main conclusions that we've made as we started, doing these kinetic studies on these enzymes was that it seemed that one of the main things that's driving the function in these systems are actually how the enzyme and substrate complex moves around in space. And so now I want to talk a little bit about how we're using mass spectrometry to study these protein motions. And so these are, I'll refer to this as the structural dynamics of the protein system. And so one of the techniques we use is called hydrogen deuterium exchange mass spectrometry. And the way that this technique works is that you take your protein and you dilute it into solvents that contain heavy water, so deuterium instead of protons. And the idea is that when you incubate the enzyme with a different substrate, or you make a mutation to the enzyme, this introduces a perturbation in the system that changes how the enzyme moves around in time. And the way that this assay works is that as this protein samples different conformations, this affects the rate at which functional groups in the protein take up deuterium from solvent. And because deuterium is heavier than protons, we can then use mass spectrometry to follow the deuterium incorporation as a function of time and learn something about how the protein is moving around. So what we do is we incubate the protein in heavy water. We then chop the protein up into hundreds of tiny peptides, and we use our MS platform to separate these peptides, both in a chromatography step. We can separate them in M/Z space, and we can also separate them in the gas phase on the basis of shape. And so we can identify many hundreds of peptides. We can quantify the deuterium uptake into all of those peptides, and then we can map those peptides onto the structure to kind of figure out where and how the protein is moving on a very localized scale. And so using data like this, we've been able to identify binding sites for the substrate, and we've been able to understand how protein motions are coordinated across this multifunctional enzyme by ligand binding, for example. And so how can we use this information moving forward? One of the main things that I want to point out is that this enzyme we study is part of a very functionally diverse family. These proteins are all related to each other, but yet they have slightly different functional properties. For example, there are some enzymes that are very specific. They'll take one peptide substrate and make one product, and they'll do this very efficiently. Whereas other enzymes in this family will take libraries of substrates and make libraries of products. And what our hypothesis is, and our initial data is supporting this, is that these different enzymes with these different properties sample motions in fundamentally different ways. And so our idea is to sort of understand how movement in these systems, enzyme systems, is related to their catalytic properties. And once we understand that, can we then use this information to engineer or alter the function of these enzymes? And this is, again, important because most of these types of enzymes make natural products that have antimicrobial or other activities that we're interested in. And then finally, we're just about to publish a paper on this. We can also use our mass spectrometry to study the overall structure of the enzyme in the gas phase. We can ionize folded proteins using very gentle conditions, and we can keep these proteins folded as they're in the gas phase, and we can study their structural properties in the gas phase. And this is just showing information, this is showing data here that suggests that our enzyme exists in two major populations. We can separate these populations on the basis of their shape. And then from this data, we can calculate structural parameters and relate those back to high resolution structures or models if we have them. And so we're using overall a set of mass spec techniques to measure the regiochemistry of post-translational modifications, and we're using mass spec to study how biomolecules move around in time and space. And so our idea is to integrate all of these methods of measurement to build very detailed biochemical and biophysical models for these biosynthetic enzymes that we can then hopefully use that information to engineer their functions in the future. So that's all that I have. I don't think I have a thank you slide, but I'm done. And as usual, I've gone a little over, sorry.
Thanks, Chris, for the interesting talk. And now let's move on to our next speaker, who is Professor Yves Lee from the Department of Physics.
All right, hello everyone. I'm Professor Yves Lee from Physics, and thank you, Axel, for the great introduction and for everyone for organizing this. So I'm a theoretical astrophysicist and I study how planets and stars form with a particular interest in exoplanets, which are planets outside of our solar system. So I use physics to answer questions like, why do most exoplanetary systems look nothing like our own? So far, we have more than 4,000 planets that we have been detected, and the number is only growing as we speak. And this is how they look like when you plot their measured masses versus orbital periods, which just means how close they are to the central star. And if you just compare this to the solar system planets, this one over here, that's our own planet, that's Earth. And you will see that they don't align at all. And in particular, there's a huge diversity in the kinds of planets outside our own. And a lot of these planets are very, very close to the central star, which is not what we see in our own solar system. In fact, these exoplanets are so alien that we give them names like hot Jupiters and super Earths. And in our own galaxy, it's these super Earths that really dominate the total exoplanetary population. And that's a big mystery because these planets do not exist in the solar system. So this event is for you guys, undergraduate students. So I thought I would just give some highlights to the projects that have been undertaken by undergraduate students with me. So this is one that's done by Jonathan Lin, who was an undergrad at UC Berkeley at the time. He's now doing his PhD at UCLA in physics and astronomy. And he did this pretty complicated calculation where he was searching for the right birth conditions for giant planets like Jupiter's and Saturn's. So planets are born in a disk of gas and dust around the star. And inside this disk, the gas gets viscously diffused away, while the solids, like the dust grains, they are drifted in towards the central star due to the aerodynamic drag, which is the same physics that describes why the feathers will take such a long time to drop to the floor if you just drop them from the top of your building. And this aerodynamic drag also helps to create planets like our own Earth, all the way from a small rocky objects like the moon. So I often get asked questions like, so you're a theoretical astrophysicist, what kind of tools do you use? What exactly is involved in theoretical research? So I use a lot of pen and paper calculations as well as numerical calculations, which means just coding. This particular paper had 49 equations in total, many of them derived by Jonathan himself. There's one example over there. But he also did a lot of numerical solving of systems of coupled ordinary differential equations and partial differential equations to evolve the underlying disk as well as the growing planets. And it has been published 2 years ago. And another project that I want to highlight is one done by our own undergraduate student, Nicholas Connors at McGill through Physics 396, which is a semester-long research class. And his goal was trying to derive the underlying core mass distribution of the exoplanets. So if you look at a planet, and if you just want to simplify it to at the very basic level, it's basically a rock with the gaseous atmosphere on top, just like our own Earth. And of course, if you talk to geophysicists, it's a lot more complicated. But if you really want to simplify it, that's basically it. And how thick those envelopes are, how thick those atmospheres are, it determines how large these planets are going to look like when you measure them. So this is the distribution of their measured radii. You'll see that it looks almost like a bimodal distribution. So this side of the radius valley, those are basically the super Earths. This side of the radius valley, those are basically the mini Neptunes. And these all together, they dominate the total exoplanetary population. However, we actually don't know whether these very, very, very small planets, smaller than the Earth, like how numerous they are, because we do not have the technical capabilities to detect them right now. However, as theorists, we can actually devise a prediction for the kind of mass distributions. So that's what Nicholas was doing. And what is the basic physics here? The basic physics here is gas thermodynamics, because how thick these envelopes can get and how much of the envelope you can retain against the high energy photons that are being thrown by the central star is all determined by the first principle thermodynamics. And there's an upcoming project for this year. This is for Physics 459, which is honors research thesis. And the goal here is to get a sense of what is this physical link between the central star and the planetary populations. So if you look at the number of planets per star as a function of orbital period, again, how close the planets are to the central star, and if you break it down into different types of the stars, you'll see that FGK, and I put in FGK and M. For those of you who are not familiar with the astronomical terms, all this means is just sun and stars smaller and cooler than the sun. And the super Earths really prefer the smaller and cooler stars, whereas the big planets like Jupiter's prefer more massive and hotter stars. And there's no theory out there that can convincingly explain why that is the case. So if you're interested in this kind of work or anything about exoplanets, come talk to me or email me at eve@physics. Thank you.
Thanks a lot, Eve. Okay, our next speaker will be Lisa Dang from Physics Matters and Astro McGill Outreach Group.
Hi, everyone. Thanks, Axel, for the introduction. So I'm also a PhD student in the physics department, where my focus, or the focus of my research, is very similar to those of Professor Lee. I study exoplanets and their climates. But today, unlike the presenters before me, I'm not here to talk about my research, but rather about what I do when I'm not doing research. So in my free time, I do a lot of outreach, and I'm a member of these two outreach groups, Physics Matters and Astro McGill. So they are the two outreach branch of the physics department, one that is more focused on general physics and the other one that is a little bit more focused on astronomy. So I just wanted to talk about the different activities that we do, and I've categorized them into 3 different categories. So we do a lot of public events, usually, or rather pre-COVID. So we do these like monthly public lectures that usually attracts a lot of people. So we try to have a good mixture of local researcher in our department to talk about what they do, but sometimes we also invite high-profile researcher from all over the world to come and talk about their own research, such as the image here is, it was an event that we had last year where Shep Dolman was coming here to talk about the famous black hole image that you might have seen around. We also sometimes do observing nights where we take some of our telescopes out and then we invite people to join us and look at different astronomical objects. We also have this recurring event that's called Astronomy on Tap. So this is a worldwide event now where we go into pubs, talk about astronomies, and have little trivia nights. Since COVID, though, a lot of our events have moved online. And so now, rather than having public lectures, we have a lot of live streams with different researchers from our department this time talking. So Professor Lee just gave one not too long ago where she talked about her life as a theoretical physicist. Upcoming this Friday, we also have another event. So if you're free and you're keen on learning about comet, I don't know if a lot of you know, but there is the Neowise comet is currently visible from Montreal. So if you join us Friday, we will give you tips and tricks on how to see this object from Montreal and from a little outside of Montreal, and also like chat a little bit about what comets are and have a little trivia night. Other than public events, we also have school visits. And so our outreach group has spent a lot of years developing a lot of hands-on activities for kids to explain very basic physics phenomenon. So we do this in multiple ways. So we used to go to schools and have kind of like a Space Explorers program. So we would go recurrently and teach them different missions that they had to complete. And each of these missions we're teaching them a concept in physics. And so, for example, in this image here, you have little kids throwing little pebbles at a random object under the circle here. And so from the collision, they have to guess which object or the shape of the object underneath. We also do workshops for elementary school and high school teachers. And so most of the time we would arrange with a school board and go to the school and give them an entire day where the teachers can ask us anything that they don't currently understand about physics. Moving forward, though, in the fall, we will be moving most of those online. And so we will be partnering up with a bunch of schools to offer these. And then lastly, we have another special type of event that is a little bit less outreach, but rather a good bridge between academia and the industry. So we organize a yearly McGill Physics Hackathon, which is not your typical hackathon. This one is science-based or science-themed. So students are invited to come and join us for a weekend of coding, where they can either learn to code, or if they are very keen already, then they can find a project and try to complete it in a weekend and present it. Unfortunately, because of COVID, we will be moving our next hackathon online to accommodate for all the students that won't be coming back in town. And I think that is it. So if you're keen in joining us, helping organizing events, or joining the outreach team, feel free to message us at outreach@physics.mcgill.ca. But also, if you're just keen in knowing what the events that we offer are, you can also message us and tell us that you're not interested in volunteering, but interested in participating and knowing about our events.
Okay, thanks a lot, Lisa. And now it's already time for our last speaker of the day, who's going to be Professor Wayne Pollard of the Department of Geography.
Thank you, Axel. I'm an Arctic geomorphologist, and basically what an Arctic geomorphologist does is studies the dynamics of landscapes and landforms and processes active in these environments. I'm not sure what's going on. Okay, so in the Arctic these days, what we hear a lot about is the impact of Arctic warming, global warming, and the fact that the Arctic is warming twice as fast as the rest of the Earth. So from that perspective, we're very aware of the fact that landscapes are dramatically changing. And we see this also in the change in glaciers and change in sea ice distribution and so on. So basically, what we need to do is recognize what aspect of landscape change is related to the current trend in global warming, and what aspects of climate change are related to the ongoing evolution of the landscape. Given that we've only just come out of a global glaciation, in the Arctic, the glaciers only receded in terms of the continental scale glaciation 10,000 years ago. And 10,000 years is a very short period of time in terms of landscape dynamics. So following permafrost, permafrost is basically ground that remains below 0 degrees centigrade continuously for long periods of time. And one of the features of permafrost is the presence of extensive bodies of ice. One of the places where I work is in the very high Arctic, and I'll show you that in a moment. But in this environment, the permafrost is very thick. It's on the order of 500 meters. It also contains vast amounts of ice stored within the permafrost that are far in excess of the normal soil moisture contents. This is a part of a long-term study. And in this talk, what I want to highlight are two unusual summers which triggered widespread erosion in this part of the Arctic. A couple of features I need to draw your attention to in these slides is, first of all, this is all extensive bodies of pure ice. And in this picture, we see a very thin layer of soil over top of the ice. This thin layer of soil will seasonally freeze and thaw. This thin layer of soil is called the active layer. The active layer buffers the underlying permafrost and the ground ice from the warm summer temperatures. So basically what we do when we study the dynamics of permafrost change in the summer is we want to analyze changing surface temperatures and how these temperatures propagate through the soil profile. So where this study is taking place is in the high Arctic on Ellesmere Island, a place called the Fosheim Peninsula, and there's a federal weather a federal government weather station there called the Eureka Weather Station. And so basically, from this point of view, the area has extremely cold conditions. We're at approximately 80 degrees north. The mean annual air temperature at Eureka is on the scale of minus 19.7 degrees. The active layer is only on the order of 60, 70 centimeters. It's a polar desert. Precipitation is less than 100 millimeters. Permafrost is more than 500 meters thick. And there's widespread ground ice. And in the map, we show a highlighted area, which is an extensive area where ice is present in extremely large volumes. In order to analyze the changes in the landscape in a process called thermokarst. Thermokarst is essentially the erosion of landscapes by melting ground ice. And so we've been looking at the trends in summer climate, because obviously that's what drives the summer dynamics. And so we use a number of variables like thawing degree days, mean July temperatures. In the summer of 2011 and 2012, we had an extremely high level of thermokarst activity. All these features here are recent examples of where the ice has become unstable and the landscape is starting to erode. And so in this case, we have several hundred new erosional features within one summer starting to form. And just for your edification, this is where the weather station is located. So basically, over the term of this, the period of this study, we've monitored well in excess of 850 erosional sites. But in 2012 alone, there was 240 new features that formed. And again, to give you a sense of the extent of the ice, here's a very thick exposure with a person standing on top. So essentially, this study is looking at how landscapes are changing in response to climate change and trying to separate the climate change-driven responses from the natural evolution of the landscape. And the natural evolution of this landscape is quite extensive. There's sea level change, there's vegetation change, there's a wide range of things that are constantly going on that will affect the landscape. So the main challenge in this study is trying to figure out what global warming is doing to this landscape. Thank you.